So then we're going to talk about, not about DNA, but rather about the proteins that are encoded by our genes. And I want to challenge us all to think about these proteins as tiny molecular machines, with the idea that understanding their structure help us understand how they function both in health and disease. So I'm going to start right away with I think I am. Well, we'll see if this goes. We'll start right away with an animation of something that's really fundamental to what we do. Actually, can I get the front lights, Jim? Maybe I can do that here. Oh, that's beautiful. Thank you. So I want to look at DNA replication in a way that we don't tend to see it. And on the left, I've thrown up some of the genes and conditions that are related to this process. But really, I want to focus on this animation and take a look at what it looks from a protein perspective. So as double-stranded DNA comes into this complex, it first encounters this nuclease, which is spinning around at 50 to 100,000 RPM. And so that molecule, instead of be spinning as fast as a jet engine, separates these two strands of DNA. The leading strand comes down below, where it encounters polymerase that is anchored to this complex. You can see the the synthesis of this continuous strand just off to the right. The lagging strands are more complicated. The complex reaches down, grabs a handful, passes it off to another chunk of the complex. That allows for 5' to 3' Saki fragment. So, I mean, this is a really crazy image, right? And it's critical to what we do in the molecular lab because there's what happen here are really what we study. But in general, I just think these animations, they're beautiful, they're exciting, they teach us something new about the process. But then some of you may be asking yourselves, sure, Eric, that's all well and good, but really for discovery and diagnosis? What is an animation? I like to think of animations as spatial modeling over time. And today I'm going to talk about some animations that include the time element and some that are just simple 3D images. I'm going to start by talking about microscopy and specifically modern advances that allow us to see across the biological spectrum. And then I'm going to continue to talk about how this aids our discovery. First, looking at specific cellular processes, teaching them apart. Second, looking at structural proteomics. And finally, drug discovery. And then at the end, I'm going to bring it all back home and talk about how it affects what we do here. So there are two real core principles that are important for visualizing molecular machines. The first is seeing things that are small, and the second is knowing what you're looking at. And so with those two ideas in mind, the real advances in microscopy started when Ernst in the late 1800s really defined the size principle. He recognized that any particle you're looking at in a microscope displays what's called a point spread function, meaning a certain amount of light and a Poisson distribution. And so when one of those Poisson distributions approaches another, slowly they become so close that you can no longer resolve them independently. And this is called the diffraction limit of light. And the model is based on this simple formula where the wavelength of light is lambda divided by 2 times the numerical aperture of the microscope. So that really gets you to the numerical aperture of the microscope with the representation quality of lenses. And so if we just keep walking through the simple exercise of saying average wavelength of light is approximately 500 nanometers, and in the best case scenario, the ideal microscope would have a numerical aperture of 1. And so with that, we reach a value of 250 nanometers. And with that, first aid defines the problems we face today. That is that diffraction limited light microscopy, we can only see down 250 nanometers. So with that scale, we're not going to be able to see any of our molecular machines. And if that were the end of the story, then this would be the last slide. But luckily, might take the volume just a little bit. I feel like I'm holding back my voice. But luckily, we found ways to overcome that. So We'll come back to size and speak to identity for a second. And this is really credit to Albert Greenwood, who in 1940 established the first immunofluorescent microscopy. That he took a fluorescent molecule and conjugated it to antibodies and found that he could specifically label proteins, and with that label structures and label cell types. That's been advanced upon in a number of ways. One extremely important way is that of using green fluorescent protein or other fluorescent protein. It was first really pioneered by Osamu Shimomura, who spent decades studying jellyfish. Like one of the most trivial scientific endeavors. He established that proteins from jellyfish fluoresce, and then Roger Chen modified those proteins to optimize them for use in microscopy. And finally, Marty Chaupy recognized that these are proteins. They could be encoded, that are encoded by genes. They can be expressed in living organisms, and completed that work. And now we have really some amazing tools in biology to express fluorescent molecules in many, many ways. One example here is that of the rainbow, where you're looking at different neurons colored by different fluorophores, all encoded by a gene expressed on the mouse. And then visualize this process here. And so now we have the world's ideal microscopes. We have a huge battery of fluorescent molecules, and we're still faced with Abe's diffraction limited challenge. And so we want to be able to image the full biological spectrum all the way down to atomic single molecules, atomic resolution of single molecules. But our diffraction limit ends here. So it's worth noting that this is the point at which we no longer resolve 2 points in space. I'm not trying to say that you can see mitochondria in this. So this is where realistically light microscopy gets down to approximately 1 micron. And for the last 15 or 20 years, there's been an arms race to break this diffraction limit and to use light microscopy to see things at better resolution. The winners of that arms race, really Stephen Pell, Bill Warner, Eric Betzig. And the first technology I'll walk you through is called stochastic optical reconstruction microscopy. And so the images I'm showing here are from Eric Betzig's lab. This is of a lysosome, or a cluster of lysosomes. And with traditional confocal microscopy, you make out the outer membrane of the lysosomes fairly clearly. And you might even say that this is the better looking image, but I'm afraid it's not. With STORM, you can see the membrane in much more specific detail. And if you zoom in, you can see these tiny particles are very close together, recognizing that this error, or this scale bar is 200 nanometers. Clearly are breaking that diffraction limit with these images. So how does this work? Cartoon down below, each molecule that's emitting fluorescence is marked in yellow. Each of those yellow molecules, it displays a point spread function in red, and they all blend together in confocal. So the trick here is they use specific photoactivatable dyes, such that when you apply light to activate those dyes, and then you can image them. And if you apply suboptimal activation, then you achieve what's called the stochastic activation, where randomly one molecule will turn on, then another, then another. When a single molecule turns on, any computer can recognize it. It can look at its large point spread function, calculate the centroid of that point spread function, place it in space precisely. Not only that, these molecules are not only photoactivatable, but they're also photobleached immediately after activation. So they turn on and they're off for the rest of the experiment. And so with this technology, you're able to light up one molecule at a time, ultimately getting the exact location of every single molecule in the sample. And so this is a great technology, but it does have its limitations. Imagine it's extremely slow, and so you can't have any living tissue involved in this microscopy setup. That's where Stephen Pell comes in. This work uses a different approach, and you're able to get images from living samples. It happens much faster. His trick, I must admit, I don't completely understand how they do this in microscopes, But the main objective is they take your point spread function, which is broad, and with a depletion beam, so this or a subtraction beam, it comes on and it takes that broad point spread function and depletes the flanks such that only the narrow spot is illuminated in the end. So without going into more detail about how that works, you're able to scan across the sample at much faster speed and image living samples. Okay, so light microscopy has now been extended down the biological spectrum to super resolution microscopy, but we're still nowhere near imaging the molecular machines that I showed you at the beginning of the talk. And so with that, we need to get away from light completely and move on to other forms of illumination. So the first being electron microscopy. Why electron microscopy? Well, first, the electron wavelength is far shorter, 100,000 times shorter than that of visible light. And so, whereas when you image a cell and look at the Golgi stacks with a confocal microscope, you see these dots that represent the entire Golgi apparatus, but you cannot hope to make out the individual cis trans Golgi stacks. And so that's what we can see here with EM. The first EM was achieved in the 1920s, and they used ultra-thin sections to visualize these very small structures. But if you've looked at much EM in your life, it can be extremely frustrating. It's as though the people in the field see things that the common person can't even see. And in looking at these pictures, they're really not very pretty at all. But you really can make out things that otherwise you can't see. And building on that, just in the last 10 years, they've now used new tilting 3D reconstruction. So what they do is they take a thicker sample and they tilt it, and by tilting it, somehow they're able to reconstruct these much more sophisticated three-dimensional structures, which is pretty cool. Okay, so that's EM, moves us down the spectrum a little bit, but really the working force of structural biology is X-ray crystallography. I think people in this room will think about X-ray crystallography, and they'll think about the work of Franklin Wilkins on solving the structure of DNA. And they did this work only decades following the discovery of this technology by Bragg 15. And so the way this technology works is you have an X-ray beam, and you shine it at a protein crystal. So now we're not looking at a biological sample, but rather we're taking a pure source of protein and throwing it in a crystal where it actually forms a uniform matrix or a lattice of proteins all combined together to form a crystal that will diffract light in a uniform way. And by taking that diffraction pattern and using, again, computational power to kind of piece together a sort of computational learning to figure out what's going on in this diffraction pattern, as you just call it computational additive. You can make a density, an electron density map, and then build a protein model. And so this technology has won over a dozen Nobel Prizes, and it accounts for 90% of the protein structures that we see when we go into places like the protein lab. And so X-ray crystallography has been a huge powerhouse in this field. But in addition to that, NMR, mass spec, circular diachroism, and some other biological tools and techniques have built onto smaller molecules. But what's the problem is we have this middle area that we really can't capture well, in addition to the limitations of what's meant in naturally limiting X-ray crystallography. And so in the last 10 years or so, this gap has been filled. It's been filled with cryo-electron microscopy. So that's repurposing old school EM towards looking at single molecules. And I think this discovery is going to have huge impact on even what we do. And I'm not the only one who thinks that. So does Nature. They published this article in 2015. I'm usually just talking about Nature as far as keeping up with something what's cool in science. But the message being that the revolution is cryo-EM. It's the future is not crystal. And so I went to PubMed to look to see if they're right. And it does look like they're right. So this is just a PubMed hits with X-ray crystallography in the title of our abstract. And it's pretty remarkable over the last couple of years how it's starting to come down. And likewise, it's pretty remarkable if you search cryo-EM that it's starting to come up. You might say, oh, these numbers are pretty low. We're only getting 400 odd publications in 2017. It may have to do with the fact that each one of these instruments costs over $5 million. So we're looking at a room which has in excess of $10 million in microscopy equipment. This is a pretty severe investment. So how does this work? Electron source, same pattern. You'll see the electron source sending some sort of particle with electrons through an embedded, your sample was embedded in vitreous ice, and then ultimately showing a pattern or a 2D image at a photodetector. This is a different way of sort of visualizing the same thing. And what I like this image is that it shows how you might see these particles. So they're very complex particles. And depending on how that particle is physically spaced, you'll see a different shadow. The people to be credited for this work are Dubichet, Frank, and Henderson, who were awarded the Nobel Prize this last year in chemistry. And just one molecule that they determined the molecular structure for is beta-lactosidase, molecule people in this room are interested in because of its role in GM1 gangliosidosis and MPS scores. And so their main breakthroughs included changing the sample preparation to immobilizing these particles in vitreous ice called cryovitrification, and then also advanced computational alignment so that they rotate and spatially average all these different single, essentially silhouettes of the protein. The computer does that for them until it spits out some very, very elaborate molecular 3D image. And finally, this improved instrumentation. And so with that, we've got images that we can now look at, like this one here. I really should have put up all the diseases this molecule is associated with, but this is the vascular ATPase. It utilizes ATP in order to pass protons across membranes and acidify compartments. So it acidifies DER, it acidifies lysosomes, it acidifies synaptic vesicles, transport vesicles. All of those things important in maintaining function of the compartments. Has over a dozen, maybe 13 to 14 different subunits, depending on which specific molecule we're talking about. And you can see here these images as they are dramatically mobile at the spin of the protein function. And so it was only with the advent of cryo-EM that we're able to see this molecule and piece it together in a whole fashion and understand how it is. So I think that's a pretty remarkable advance. Okay, so cryo-EM allows us to look at samples in their native state. It's very good at looking at large molecules, and you can get near atomic resolution extremely fast and with limited sample preparation. That's in contrast with that of X-ray crystallography, where really the bottleneck is getting crystals to grow. I know structural biologists who talk about their PhDs pretty much spending all of their time trying to get crystals. It's pretty amazing testing. And once you do have a crystal and you get a structure, it's really hindered by the problem that the crystal itself may be modifying the structure of the protein. So that's concern. That's not concerned with cryo-EM. Okay, so I think now we have our whole biological spectrum illuminated, and what can we do with this information? In the next part of my talk, I want to talk about how understanding these structures and modeling them specifically aids discovery. And if you're at all skeptical about that, I want you to rethink this story a little bit, because we don't think of Wilkins and Franklin, or any of us do, but the popular media doesn't think of Wilkins and Franklin as those that discovered the structure of DNA. The credit went really to Watson and Crick. They didn't generate the data. They weren't the brilliant structural biologists, but they were willing to take the data, put it in a new space and manipulate it in creative ways to solve the problem. And I really think that that's what animation and modeling is doing for some of our current problems. So as an example of that, I want to turn to the issue of making a vesicle. So taking a planar lipid bilayer and gathering it up into a spherical vesicle. And I'm going to go through a model or an animation that describes this process called platinum-mediated endocytosis. And it's important to think about in making a model like this, consider the cellular context, the protein structures that are involved, and then also the molecular interactions that are involved. A lot of times these interactions are derived from biochemical. We're going to start with just a video using some super resolution microscopy in a technique that I actually didn't present, which I don't know much about, it's brand new. But again, I've thrown up some genes on the left and the associated inherited conditions. And what you're seeing here is actin polymerization in the cell, colored in red, and you're seeing green spots, both large green spots and small ones that turn on and off. Those are platinum. And so I think it's a pretty incredible image, but it also illustrates that each one of these sort of data collection modalities is very limited. In this case, we just get to capture the dynamics of two molecules. And so that information can then be applied to a model. And so I've modeled it here with this cartoon. We have a flattering page and how it surrounds a vesicle from actin and how it pulls it up from the membrane. And actually, the animation I'm going to show you, that actin is gone. We're just going to focus on the flattering components. And so flattering it looks like this, called a triskelion, that has 3 legs. And this is based on structural information. And it interacts with a protein called AP2, or the AP2 adaptive protein complex. And that interacts with the transmembrane receptor. And so with that, I'll play this video. I'll turn on the sound. Okay, so AP2 is dancing across the membrane. And the clathrin triskelions randomly are bouncing in the lumen of the cell, sort of bouncing in space until they anchor down to one adaptive protein. Slowly, you accumulate more and more. And if you take those away, you can sort of see the vesicle begin to form. It's a cooperative process, and once you have a certain number, then the others come in quickly. This forms a neck where the membrane meets the plasma membrane, and that high curvature attracts what's called diamine. And diamine is a ratcheting protein. If you focus in on it, it comes in, ratchets, and actually pinches off the plasma membrane from the vesicle, pops. And so now the vesicle floats away, but importantly, it's not useful until it loses plasma. And so auxilin and HSP70 are these yellow and orange things that come in with a locking mechanism. One by one.
Here, where is this platform?
This could become
from this.
This could be popping off. So platinum-mediated endocytosis is used in many different places. This could be coming off of the plasma membrane at a synapse, so that is a synaptic vesicle, or it could be popping off the ER. So that's great. I was going to fast forward to that. So anyway, then you lose all those different platinum pieces, and the vesicle is now a naked vesicle. And so that was work from Janet Owasso and Tommy Kirkhausen. Janet Owasso was a biologist turned animator, and now she's animating for people all over the world, which is really cool because it's she sort of kept her biology roots, but she just does the work to animate problems like that. And so in making a model like this, you're forced to face questions of scale, stoichiometry, kinetics. So you need to know what size of the molecules I'm looking at, how many are they, and how do they bounce around in space based on thermodynamics. And you can apply numbers to all those things, and the most sophisticated models actually can use them. You can put it all into a computer and hit go and see what happens. And so the take-home message is when asked Connie Kirkhausen what he learned from this experiment, this sort of in silico experiment, is he said modeling these interactions, he learned that they were far weaker than he had first realized, and that in fact he noticed that what would happen is you'd have these events that would start, but then they would abort. And so they're extremely cooperative interactions. And what I think is interesting is he made that comment about these interactions starting and then aborting. And I showed you that image to begin with. In fact, the microscopy image that I showed you, or the video that I showed you, came after this animation. So in some ways, I think you really did predict that prior to the microscopy getting to that point. Okay, so that really talks about how we can discover new things about protein dynamics using animations and modeling. What about with proteomics? Again, this is something that's come up in a big way over the last approximately 10 years. And this paper, three-dimensional reconstruction of protein networks, provides insight into genetic disease, is not the first and not the only of its kind, but I think it did a very nice job of it. So what did they do? They took structural information from the protein data bank, the website, and then they took biochemical interactions, which were essentially text mined from PubMed. So they went to look through PubMed and automatically pulled in information based on these two hybrid screens or cloning of precipitation that said these two molecules interact in vitro situation. And so with that, they brought in 20,000 interactions and over 7,000 different proteins. And then they queried gene disease associations in HBMD and OMIM, and essentially brought in 60,000 different mutations, 4,000 genes, and 3,000 distinct disorders. With all this information, they simply throw it into a blender, and what comes out looks just like they threw it into a blender. But we can zoom in on it, and essentially what you see here is a whole bunch of nodes. Each one of those nodes represents a protein. The size of the node represents the number of interactions. The color of the node represents the essentially number of diseases they're associated. And so, given this interactome that they've created in computer space, they can start asking it questions. And the first thing to do is just validate it to make sure it's doing what you'd expect it to do. And so they asked, what's the chance that a missense mutation that we know is associated with disease is going to occur at an interaction interface? So a place that we know two proteins. And it turns out that they're enriched over twofold. That compared to other domains that we do not really know what they're doing, and we see no enrichment. And then if you look outside domains, so in unstructured parts of the protein, you see in black a depletion of pathogenic genes. So that's in line with what one would think. Similarly, we could ask, how about loss of function mutations, so changes that essentially result in truncation of the protein or early termination or loss of transcript? You would predict that that could happen in a domain or out of a domain, and it wouldn't matter. And that's again what they see. They see no specificity in localization of those things. So it seems the model is predicting things that we would also predict. What about novel things? So they conclude, and I'm cutting the chase here, they conclude 292 new disease genes in 182 different diseases. So I'm going to walk through the one that they expanded on in the main text of the paper. They said TP63, which if you were present at Dr. Hall's talk this week, recognize that it's involved in AEC and Rapp-Hodgkin syndrome, among others. But specifically, they were interested in mutations that clustered at the interface and mutations that were associated with these conditions. And they identified that this interface is an interaction interface with TP70. The TP73 has no disease association. And so in order to validate this, they took in vitro studies and demonstrated that these specific changes associated with these conditions specifically disrupt TP73 interaction and not other interactions, not folding of the protein. They didn't go, unfortunately, they didn't go and find patients for us with TP73 changes, but that has yet to be discovered. But that was in 2012, and interactomics is still a hot topic. So this is another paper, again in Nature, this is in 2017. saying that the goal is to develop a comprehensive human protein-protein interaction network. We're currently at 100,000 interactions, and the goal is to reach somewhere around 1 million. So we can call that a work in progress. So that brings us from protein dynamics and proteomics down to drug discovery. And these points are pretty simple. Again, we're turning back to cryo-electron microscopy. We're able to actually get atomic resolution as far as the structure of proteins that we were not able to get before. And here we've got what's called a potassium KTP channel. I'm sorry. Yeah, that's right. Potassium KTP channel sitting in a plasma membrane, which is marked by these two gray lines. Here you're looking at it in profile, and here you're looking top down. Essentially, this is the core of potassium channel. And so with this new imaging information, we can apply different drugs. I mean, for the purpose of talking about green drug and our red drug, but we can apply different drugs to the preparation before you do the imaging, and you can see exactly where those drugs bind. It is useful information because that can help you define or to engineer a better drug as far as being binding sites. But then you can also look at the conformational changes that occur when they combine. So that's that topic, or that's that specific problem as far as drug discovery. And then this is another one. So Zika virus, when it hit the Americas, was obviously all over our press, and the cryo-EM people immediately took this up as a challenge. They were able to solve the crystal, or the crystal, they were able to solve the molecular structure of the Zika virus in a matter of 3 months. So they're using it to develop better vaccines. So that kind of concludes the discovery phase of my talk. And so now, how do we take all this information and apply it to what we do? Well, most of you know that molecular lab is interested in identifying sequencing variants and then classifying those variants as whether or not they cause disease. And this is the main paper that we use to guide our classification criteria. And ultimately, we classify things as anywhere from pathogenic to benign and some place in between. And we do that based on criteria that come from protein or population databases, computational and predictive tools, functional assays, and inheritance. Everything that I've talked about today, I should say, within these categories, we then apply these criteria, which can be supporting, which is quite weak, up to very strong. So everything I've talked about today falls into this category of computational and predictive tools. And as far as how it contributes to our classification, we only get one sort of wimpy supporting piece of evidence that we can apply with those tools. So what tools do we use? We use mutation taster, SIFT and Probian, and Polyperm2. And these are really 3 and maybe 4. These are developed by and similar, but they're really some of the most the highest performance, I would say, and those that are most accepted in the field. And so together, we're able to apply supporting evidence if, but only if, they all agree. So not only do we only get one piece of supporting evidence, but if we have one outlier, then it all goes up. So I think you're getting the message that very weak support for classification. The data that's included in these includes evolutionary constraints, their position in one dimension along the length of the protein, the amino acid properties, and then polypen-2 is the only one that actually takes into consideration their position in three-dimensional structural space. And but the one limitation of polypen-2 is it doesn't include all this other data. So it doesn't include protein-protein interaction interfaces. It doesn't include binding pockets or active sites. And it doesn't include any other information as far as where those previous pathogenic mutations may cluster in three-dimensional space. So that brings us to one more thing. There's an additional piece of evidence that we may apply that does relate to this protein structure, and that is the mutation hotspot criteria. And so if we have a variant that falls in a region where we only see pathogenic change and we see no benign change, and we can apply moderate evidence, which is sufficient, which is relatively less moderate, it can contribute significantly to classification. So as an example of that, I bring up this SMAD binding protein SKI. The SKI gene has two hotspots. The red and black aren't really matter at this point, previously published. And these two hotspots, mutations here, contribute to the Spritz-Goldberg syndrome. And I bring this paper in. There are many examples of hotspots, and because this is one example of a paper that was essentially predicted by the interactome data that I showed you earlier. So they were the first to suggest this gene interacts with SMAD. Okay, so that's great, but what about when they don't cluster? In this image of KRAS, where you have mutations shown along the length of the KRAS gene, I would say these mutations do not cluster in space. But it's important to recognize this is one-dimensional space, and this paper recognized that in its own point, saying this published comprehensive assessment of cancer missense mutations clustering in protein structure. And so they looked at proteins like this, where the mutations are scattered throughout the length of the protein, but cluster in 3 dimensions. I think it's useful to look at this plot down below, that we call a surface plot. And so we orient this single one-dimensional sequence from the N-terminus to the C-terminus. The red amino acids that are in bold are recurrent pathogenic changes found in somatic tumors. And the gray lines, drawing them all together, tell you how many angstroms apart each of those are. So in this case, all of these lines connect them, and they're less than 13 angstroms apart. Really, this describes that this is an interacting interface of a. So I think this shows that pathogenic changes are occurring in these interacting interfaces. And there are other examples of this. This is from another cancer paper looking at TP53. And you can look at TP53 interacting with itself, sitting over DNA, or interacting with the protein SV40. Shares the same interaction interface. That interaction interface is highlighted in red in this plot. And this shows in black the number of mutations across the length of the protein. So this image doesn't display terribly well. There are a number of black enrichment points that occur in this interaction interface, and this is thought to be highly statistically significant mutation clustering. And so once again, this is contribute classifying variants, but moreover, it predicts the outcome in the patient. So if we look at survival, probability of survival in these patients, if you look at the green, we see mutations outside of the interacting interface in TP53. If you look at blue, this is non-TP53 mutations, so in other cancer-causing genes, but in individuals with cancer. And finally, if you look in this red line, this represents patients that have mutations that fall in the interacting interface. So not only are these mutations informing diagnosis, but potentially prognosis. So in general, the cancer field is really driving this 3D modeling piece forward. They have a lot more data to work with, and now there's new tools, new prediction tools that become available that promise to be able to localize these mutations in 3D structures. So my message in concluding this section is really our ability to predict pathogenicity based on all this information is extremely underpowered. I don't advise that we change what we're doing immediately, but I like to think that we can keep an open mind to new tools that are coming about with the ultimate goal of decreasing the VUS burden and increasing our diagnostic field. And so, in conclusion, I'll play this video. Oh, we don't need sound. I'll play this video just for fun as I walk through my. So I hope you can appreciate the new microscopy advances that have been made both in cell biology with super resolution as well as in structural biology with understanding with using cryo-EM. And this modeling these different structural proteins and their cellular space can aid for understanding dynamic molecular processes, but also for proteomics and drug development. And finally, modeling may aid diagnosis. Currently, our tools are rather rudimentary, but I believe has a bright future, and new strong tools will help us really guide our understanding forward. So now I'll take any questions.